Hello everyone, in this video series we are going to start preparing for CSIR Net Life Science stream. So this particular video is about immunology, 1 hour immunology, initial portions of immunology syllabus, life science portion. We are going to learn about the science known as immunology, and we'll be following the Pathfinder study material. So let's start. Immunology is the science that is concerned with the immune response to foreign challenges. Immunity, derived from Latin word immunis, meaning exempt, is the ability of an organism to resist infection by pathogens or state of protection against foreign organisms or substances. The array of cells, tissues, and organs which carry out this activity constitute the immune system. Immunity may be active or passive, and innate or adaptive. Active and passive immunity. Active immunity is the adaptive immunity that is induced by natural exposure to a pathogen, which is natural, or by vaccination, artificial. Active immunity is the adaptive immunity that is induced by natural exposure to a pathogen, either by natural exposure or by artificial vaccination like mechanisms. In this kind of immunity, a person develops his own immune response to a microbe. Immunity resulting from transfer of antibodies or immune cells from an immune to a non-immune individual is known as passive immunity. It may be naturally involves the transfer of antibodies from one host to another, or artificially when antibodies or lymphocytes that have been produced outside the host are introduced into a host. That is acquired one. So active immunity, exposure to antigen, immunity achieved by injecting antigens, activation of immune system, immune state develops over a period of weeks. Immunological memory develops. But in case of passive immunity, no exposure of antigen, immunity achieved by injecting AB or AG reactive T cells, no immune system activation, immunity develops immediately, no immunological memory develops in case of passive immunity. Next is about innate immunity. Innate in the sense non-specific or natural immunity is present from birth and consists of many factors that are relatively non-specific. That is, it operates against almost any foreign molecules and pathogens. It provides first line of defense against pathogens. It is not specific to any one pathogen, but rather acts It acts against all foreign molecules and pathogens. It also does not rely on previous exposure to a pathogen, and response is functional since birth and has no memory. In elements of innate immunity, physical barriers. Physical barriers are the first line of defense against microorganisms. It includes skin and mucous membrane. Most organisms and foreign substances cannot penetrate intact skin, but can enter the body if the skin is damaged. Secondly, the acidic pH of sweat and sebaceous secretions, and the presence of various fatty acids and hydraulic enzymes like lysozyme inhibits the growth of most microorganisms. Similarly, respiratory and gastrointestinal tracts are lined by mucous membranes. Mucous membranes entraps foreign microorganisms. The respiratory tract is also covered by cilia, hair-like projections of the epithelial cell membranes. The synchronous movement of cilia propels mucus entrapped microorganisms into these tracts. Similarly, the conjunctiva is a specialized mucus secreting epithelial membrane that lines from the interior surface of each eyelid. It is kept moist by the continuous flushing action of tears, lacrimal fluid, from the lacrimal glands. Tears contain lysozyme, lactoferrin, IgA, and thus provide chemical as well as physical protection. Microorganisms do occasionally reach the epithelial barricades. It is then up to the innate and adaptive immune systems to recognize and destroy them without harming the host. In case of innate immune response, several antimicrobial chemicals and phagocytic cells provide protection against pathogens. Then chemical mediator. A variety of chemicals mediate protection against microbes during the period before adaptive immunity develops. The molecules of the innate immunity system include complement, cytokines, pattern recognition molecules, acute phase proteins, cationic peptides, enzyme like lysozyme, and many others. What is complement? Complement is a heat labile component of blood plasma that augments phagocytosis. So, what is a complement? Complement is a heat labile component of blood plasma that augments phagocytosis. This activity was said to complement the antibacterial activity of antibody, hence the name complement. It also bridges innate and adaptive immunity and removes immune complexes. The complement system is composed of over 30 serum proteins. Activation of complements in response to certain microorganisms results in a controlled enzymatic cascade which targets the membrane of pathogenic organisms and leads to their destruction. Then cytokines. The term cytokine is a generic term for any low molecular weight. The term cytokine is a generic term for any low molecular weight soluble protein or glycoprotein released by one cell population which acts as an intercellular mediator. It includes monokines, lymphokines, interleukins, interferons, and others. Cytokines are required for immunoregulation of both innate as well as adaptive immune response. Interferons are cytokines made by cells in response to virus infection. Remember, interferons are also cytokines, but they are specific to virus infection. Interferons are cytokines made by cells in response to virus infection, which essentially induce a generalized antiviral state in surrounding cells. Then chemokines. Chemokines are small, positively charged, secreted proteins that have a central role in guiding the migrations of various types of white blood cells. Cytokines are small, positively charged secreted proteins that have a central role in guiding the migrations of various types of white blood cells. They bind to the surface of endothelial cells and to negatively charged proteoglycans of the extracellular matrix in organs. We just mentioned cytokines are positively charged, so they bind to negatively charged proteoglycans of the extracellular matrix of organs by binding to G protein linked receptors in the surface of specific blood cells. Cytokines attract the cells from the bloodstream into an organ and then help stop migration. And next topic, pattern recognition molecule. Many molecules involved in innate immunity have the ability to recognize a given class of molecule. Recognize pattern. Patterns are conserved structures and invariant among microorganisms of a given class. Pattern recognition molecule that recognize pathogen-associated molecular pattern PAMP may be soluble, circulating proteins, or cell surface receptors. Many PAMPs are recognized by pattern recognition molecule present on the surface of phagocytic cells. Then mannose binding lectin, mannose binding lectin MBL, and C-reactive protein CRP are soluble protein recognition molecules that bind to microbial surfaces and promote their opsonization. Toll-like receptors or TLRs are a class of pattern recognition molecule that function exclusively as signaling receptors. There are at least 10 distinct proteins in this family of mammalian receptors. For example, toll-like receptor 4 signals the presence of bacterial lipopolysaccharide or LPS and heat shock proteins. So TLR2 signals the presence of bacterial lipoproteins and peptidoglycans. Then acute phase proteins are a heterogeneous group of plasma proteins mainly produced in the liver as the result of a microbial stimulus. They include C-reaction protein, CRP, serum amyloid protein A, serum amyloid protein A, SAA, and mannose binding protein, MBP. Cytokines IL-1, IL-6, IL-8, etc. released by macrophages upon activation by bacteria stimulate the liver to rapidly produce acute phase proteins. These proteins maximize activation of the complement system and opsonization of the invading microbes. Then cellular defenses. The cells responsible for both non-specific and specific immunity are the leukocytes. Many specialized cell types like neutrophils, macrophages, monocytes, NK cells participate in innate host defense mechanisms. Once a pathogen evades the physical and chemical barriers, the specialized cells play important role in protection. Phagocytosis is a fundamental protective mechanism carried out by these cell types: neutrophils, macrophages, monocytes, and dendritic cells. Phagocytosis is the ingestion of invading foreign particles such as bacteria by individual cell. Phagocytosis may be enhanced by a variety of factors collectively referred to as opsonins, Greek word meaning prepared food for, consist of antibodies and various serum components of complement. The process by which particulate antigens are rendered more susceptible to phagocytosis is called opsonization. After ingestion, the foreign particle is entrapped on a phagocytic vacuole, phagosome, which fuses with the lysosome, forming the phagolysosome. The antimicrobial and cytotoxic substances present within the lysosome destroy the phagocytosed microorganism in following ways. First, oxygen-dependent killing mechanisms. During phagocytosis, a metabolic process known as the respiratory burst occurs in activated phagocytes. It results in transient increase in oxygen consumption by cell. Activated transient increase in oxygen consumption by cell. So during phagocytosis, a metabolic process known as the respiratory burst occurs in activated phagocytes. It results in transient increase in oxygen consumption by cell. Activated phagocytes generate a number of toxic products like reactive oxygen intermediates such as hydroxyl radicals, hypochlorite anion, superoxide anions, hydrogen peroxide, and reactive nitrogen intermediates like NO, NO2, HNO2 that have potent antimicrobial activity. And oxygen independent killing mechanisms. Activated macrophages also synthesize lysozyme defensins, cysteine-rich cationic peptides containing 23 to 35 amino acid residues and various hydrolytic enzymes, cytotoxic peptides whose degradative activities do not require oxygen. Then inflammatory barriers. Inflammation is an important non-specific defense reaction to cell injury. The hallmark signs of inflammation are pain, redness or erythema, and swelling, edema, and heat. Each of these are the result of specific changes in the local blood vessels. Pain is caused by increased vascular diameter, which leads to increased blood flow, thereby causing heat and redness in the area. The blood vessels become permeable to fluid and proteins, leading to local swelling and an accumulation of blood proteins that aid in defense. At the same time, the endothelial cells lining the local blood vessels are stimulated to express cell adhesion proteins that facilitate the attachment and extravasation movement of blood cells through the vessel wall into the surrounding tissue of white blood cells, including neutrophils, lymphocytes, and monocytes. The inflammatory response is mediated by a variety of signaling molecules. Activated macrophages produce chemoattractants known as chemokines. Many of these attract neutrophils, which are the first cells recruited in large numbers to the site of the new infection. Others later attract monocytes and dendritic cells. The dendritic cells pick up antigens from the invading pathogens and carry them to nearby lymph nodes where they present the antigens to lymphocytes to marshal the forces of the adaptive immune system. Two principal mediators of inflammatory responses are histamine, released by a variety of cells in response to tissue injury, and kinins, present in blood plasma in an inactive form. Both cause vasodilation and increased permeability of capillaries. Kinins are also very potent nerve stimulators and are the molecules most responsible for pain associated with inflammation. Here we could see a question related to this topic. How might an arthropod such as a cockroach or beetle protect itself from infection? In what ways might be the innate immune response of an arthropod be similar to those of a plant, and how might they differ? So, invertebrate animals may protect themselves against environmental pathogens with simple forms of innate immunity. For example, the fruit fly produces peptides with antifungal activity. These small molecules protect the insect from a fungal pathogen commonly encountered in its environment. Plant immunity is similar in the plants also produce antimicrobial peptides and a host of their agents such as oxidizing radicals and enzymes that attack microbial cell walls. One of the many differences between plant and arthropod immunity is that plants lack phagocytic responses which are present in arthropods. Next, adaptive. Adaptive in the sense specific immunity. Adaptive immunity, also known as acquired immunity or specific immunity, is capable of recognizing and selectively eliminating specific foreign antigens. It does not come into play until there is an antigenic challenge to the organism. Adaptive immunity display four characteristic features. First one, antigenic specificity. It is the ability to discriminate among different epitopes or antigens. That is antigenic specificity. Then immunologic memory. This is the ability to recall previous contact with a foreign molecule and respond to it in a learned manner, that is, with a more rapid and large response. Then diversity, the ability to respond to many different epitopes, even if the individual has not previously encountered them. Diversity, the ability to respond to many different epitopes, even if the individual has not previously encountered them. That is diversity. Then self or non-self recognition. It is the ability to recognize and respond to molecules that are foreign and to avoid making a response to those molecules that are self. Difference between the innate and adaptive immune response. Innate, antigen non-specific, rapid response, hours, no memory. But adaptive immunity, antigen specific, slow response, days. Memory response to repeated infection is identical to primary response. Next, immune responses of adaptive immunity. There are two arms or branches of acquired immunity which have different sets of participants and different purposes, but one common aim to eliminate the antigen. These two arms interact with each other and collaborate to achieve the final goal of eliminating the antigen. Of these two arms of the acquired immune response, one is mediated mainly by B cells and circulating antibodies. A form of immunity is referred to as humoral. The word humors was formerly used to define body fluids. Humors, just remember some of the terms we learned, which is humor, aqueous humor. That terms which are usually used to refer to body fluids. So same is the case here. The other is mediated by T cells, which do not synthesize antibodies but instead synthesize and release various cytokines that affect other cells. Hence, this arm of the acquired immune response is termed cellular or cell-mediated immunity. In humoral immunity, B lymphocytes synthesize and secrete antibodies with specificity against the foreign substance, whereas the T lymphocytes, which also exhibit specificity against the foreign substance, by virtue of their T cell receptors, TCRs, do not make antibodies but perform various effector functions when antigen presenting cells or APCs bringing antigens into the secondary lymphoid organs. T lymphocytes also interact with B cells and help the latter make antibodies. They are active macrophages and have a central role in the development and regulation of acquired immunity. Acquired immunity mediated by T lymphocytes is termed cell-mediated immunity. Then innate and adaptive immunity are not independent. The innate and adaptive immunity operate in cooperative and interdependent ways. The activation of innate immune response produces signals that stimulate and direct subsequent adaptive immune responses. They cooperate in many ways. Here I could see a flow chart which represents the relationship between innate and acquired immunity, adapted and modified from immunity, a short representation. So here we could see innate immunity divided into physical barriers, cells, chemical barriers. Physical barriers again into skin, mucous membranes. Chemical physical barriers, skin, mucous membrane. Chemical barriers, pH, lipids, enzymes, etc. Cells, pattern recognition molecules, PMNs, monocytes, macrophages, eosinophils, NK cells, cytokines, antibodies, cytokines, B cells, T cells, antigen specific receptors leading to acquired immunity. The encounter between macrophage and microbes can generate danger signals that stimulate and direct adaptive response. It may increase the ability of macrophages to display antigen for recognition by antigen specific T cells. Macrophages stimulated by encounters with microbes also create immunoregulatory molecules called cytokines. These cytokines guide adaptive immune response. Vice versa, adaptive immune system also produces signals and components which stimulate and increase the efficiency of innate response. The cells of adaptive system, T cells, secrete cytokines and increase the ability of macrophage to kill the ingested microbes. By binding to the pathogens and antibodies, mark it as a target for attack by complement. Cells of the immune system. The immune system is a defensive system in a host consisting of widely distributed cells, tissues, and organs that recognize foreign substances and microorganisms and acts to neutralize or destroy them. The cell responsible for both non-specific and specific immunity are the leukocytes or white blood cells. All of the leukocytes originate from pluripotent stem cells in the fetal liver and in the bone marrow of the animal. Pluripotent stem cells in the bone marrow divide to form two bloodline lineages. The lymphoid stem cell give rise to B cells, T cells, and natural killer cells. The common myeloid progenitor cell give rise to the granulocytes. Granulocytes in the sense neutrophils, eosinophils, basophils. Monocytes that give rise to macrophages and dendritic cells, an unknown precursor that give rise to mast cells. Megakaryocytes that produce platelets, and the erythroblast that produces RBCs. Hematopoietic stem cells present in bone marrow, myeloid progenitors and lymphoid progenitors. Lymphoid progenitors, dendritic cells, natural killer cells, B cells and T cells. Myeloid progenitor, monocyte, basophil, eosinophil, neutrophil, mast cell, platelets, RBC. Monocyte, basophil, eosinophil, neutrophil, mast cell, platelets, RBC. Once again, monocyte, basophil, eosinophil, neutrophil, mast cell, platelets, RBC. They mature into macrophage dendritic cells. First one is lymphocytes. Lymphocytes responsible for adaptive immune response are mononuclear leukocyte constitute 20 to 40% of WBC. 20 to 40% of WBC. They occur in large numbers in blood and lymph and in lymphoid organs such as the thymus, lymph nodes, spleen, and appendix. Up to 99% of lymphocytic cells are found in lymph. Lymphocytes are of three main types: B lymphocytes or B cells, T lymphocytes or T cells, natural killer cells or NK cells. First, B lymphocytes. A lymphocyte that mature in the bone marrow in birds is Bursa fabricius. If you learned about the Columba livia, that means just domestic pigeon internal structures, might have learned about Bursa fabricius. So that portion in birds in general acts as a maturation site for B lymphocyte. A lymphocyte that matures in the bone marrow and expresses membrane-bound antibody. After interacting with antigen, it differentiates into antibody-secreting plasma cells and memory cells. They are the only cell type capable of producing antibody molecules and therefore the central cellular component of humoral immune response. B cells also serve as antigen-presenting cells or EPCs. Then T lymphocytes. T lymphocytes arise in the bone marrow. Unlike B cells, which mature within the bone marrow, T cells migrate to the thymus gland to mature. During this maturation within the thymus, the T cell comes to express a unique antigen binding molecule called the T cell receptor on the membrane. T cells do not make antibodies but perform various effector functions when APC bring antigens into the secondary lymphoid organ. T cells help in eliminating APCs, cancer cells, virus infected cells, or grafts which have altered cells. T cell expresses distinct membrane molecules on the basis of presence of one or the other of two membrane molecules, CD4 plus and CD8 plus. There are two subpopulations of T cells. T helper cells carry CD4 plus membrane glycoprotein on their surfaces, and T cytotoxic T cells carry CD8 membrane glycoprotein on their surfaces. In addition, all T cell subpopulations express the T cell receptor that includes CD3. The ratio of CD4 and CD8 T cells is approximately 2:1 in normal human peripheral blood, but it may be significantly altered by immunodeficiency and autoimmune diseases. Then cytotoxic T cells provide protection against intracellular pathogens such as viruses and some bacteria and parasites that multiply in the host cell cytoplasm, where they are altered from attack by antibodies. They provide this protection by killing the infected cell before the microbes can proliferate and escape from the infected cell to infect neighboring cells. Helper T cells are crucial for defense against both extracellular and intracellular pathogens. They help stimulate B cells to make the antibodies that inactivate or eliminate extracellular pathogens and their toxic product. They also activate macrophages to destroy any intracellular pathogen multiplying within the macrophage phagosomes, and they help activate cytotoxic T cells to kill infected target cells. And natural killer cells. Natural killer cells and NK cells are a class of lymphocytes that are distinct from cytotoxic T cells. However, NK cells also play a role in destroying cells infected with intracellular pathogens. NK cells are neither T cells nor B cells. They constitute 5 to 10% of lymphocyte population. Their numbers are not enhanced, nor do they exhibit memory after stimulation. NK cells destroy the target cell not by phagocytosis but by releasing biologically potent molecules. They resemble TC cells in their ability to destroy infected cells. For example, NK cells also use perforin and granzymes to kill their targets. However, NK cells differ from TC cells in that they kill targets in the absence of a specific antigen. NK cells are capable of destroying malignant and virus-infected cells without previous exposure or contact with the foreign antigen. Shetiak Higashi syndrome. I'm not sure about the pronunciation. Just look at the spelling. Chedik. Higashi syndrome. An autosomal recessive disorder is associated with a lack of NK cells. NK cells recognize targets through two ways. In some, they imply NK cells receptors that can detect abnormalities such as low MHC1 display and unusual markers on cancer cells and cells infected by some viruses. Another way in which NK cells recognize potential target cells depends on certain viruses upon the fact that some tumor cells and cells infected by certain viruses display antigens against which the immune system has made an antibody response, so that anti-tumor or antiviral antibodies are bound to their surfaces. Because NK cells express membrane receptor CD16 for IgG, they can bind to those antibodies and subsequently destroy the targeted cells. This is an example of a process known as antibody-dependent cell-mediated cytotoxicity, or ADCC. Next topic will be dealt with the help of next video. Thank you for listening.